Here's my disclosure. I'm a founder of two companies. And when I think about how to start my talk, the only thing I can think of kind of related to the double helix discovery is I went to Stanford many years ago. My mentor is Stanley Cohen. It's my postdoc supervisor. And oftentimes, I'm kind of envy the old good time when they can basically have a conference in Hawaii, Waikiki, I think, one of the small restaurants. They can come up with a genius idea to do recombinant DNA. Even nowadays, every lab's working in molecular biology, relevant to molecular biology, still using the plasmid as the most convenient way for cloning work. So when I established my lab, actually, a few years after I started my own lab in PKU, I started to working on genome editing or gene editing. I don't remember at that time whether we have the concept of gene editing. I just want to achieve a goal which we couldn't do back then, to have a gene knockout technology developed in eukaryotic. It turned out we try many different kind of a way and a lot of effort in so-called foundation technology, and many of them actually failed. It turned out we spent much of our effort in CRISPR to develop a series of technology associated with so-called functional genomics to decode protein coding gene, non-coding RNA, 3D structure, and so on and so forth. So I decided to go in a kind of historic way or perspective to walk through what we have been doing throughout the years. So one thing actually, the most important motive for most of the scientists at that time try to get into the genome editing is to achieve gene knockout, okay? This is again the strongest point for us to get into the field. Another thing actually we're thinking about whether we can turn this into a high-throughput fashion to decode gene function in a massive kind of way. And we tried that actually in the tail stage. It couldn't work out for a number of reasons. But with the CRISPR system, it become much more convenient. Basically, it's a two-component. We can express Cas9 as the foundation, and the other component is a guide RNA, which can really make a very convenient library to dictate what kind of gene we target by changing the sequence of sgRNA, and turn out we can put this in a whole package to make a library to target a gene in a massive delivery way. So the trick actually to have this CRISPR screening methodology is to combine with the high-throughput screening technology and the bioinformatic decoding to tell us the most likely the gene whose knockout will give us the phenotypic change of our interest, okay? So this CRISPR screening has been applied for many years, obviously, in a number of labs. And one of very few applications in our hand, actually, because we devote most of the time in the technique itself, okay, this is one of the examples. We team up with Fugao and Zhengdeshi labs to understand and to study a very important viral infection problems called echovirus. So through a very simple setup for the CRISPR screening, we can decode 3 genes, CD55, FCGRT, and B2M. CD55 encodes a protein which is known for this very virus, for the virus attachment to the cell surface. FCGRT, B2M turn out to be a new player, but the two very famous genes encoding 2 proteins forming this complex receptor called FcRn. It turned out these very complex receptors has been hijacked by the very virus for its finish their life cycle. Actually, the original role for FcRn is serving as a transporter for the fetus to get the IgG in mother, okay, from mother, okay. So we continue on optimize the CRISPR screening technology, and one of the very important optimization is to increase efficiency. and sometimes to lower the cost, okay? So important setup for this kind of optimization is we embedded internal barcode in the loop region of sgRNA. By that way, we can really significantly increase the efficiency. So without getting into any of the details, basically we can have a 1 to 2 log of efficiency boost. So essentially the idea is to allow multiple sgRNA entering the same cell, okay? And we can separate the noise from the signals. But the problem to apply this kind of approach is to allow multiple sgRNA into the same cells and using the Cas9 to create double-stranded DNA breaks. Okay, that will basically lead to a certain level of cytotoxicity. To solve that kind of problems, we combine with base editor for high-throughput screening. But the only thing is we choose CBE, which is a C to T kind of a conversion base editor to do gene knockout, okay? So we tried 3 different ways to achieve that. One is to turn normal sense codon to stop codon, to generate premature stop codon. Alternatively, we can target the ATG to basically disrupt the translation initiation. And the third way to generate gene knockout strategy using base editor is to perturb splicing, okay? So through that, we basically generate a new way to do gene knockout using a base editor. And we call it the BARBEKO, okay? So it's using iBAR strategy together with BE, base editing, and achieve gene knockout strategy. So that way we can really have a very efficient way to do gene knockout in a high-throughput fashion. And even we can working on some very sensitive cell line close to primary cells, which has the p53 wild type, which is super sensitive to double-strand breaks. So besides gene protein coding gene, we are also interested in study in a high-throughput fashion, so-called non-coding RNAs. We have a number of different kinds of non-coding RNA, microRNA, siRNA, SNO-RNA, even non-coding RNA. But we don't have a very efficient way to study their function because of lacking of technology. And for the high-throughput decoding of those kind of non-coding RNA function, we just cannot come back to the original strategy for protein coding gene. The reason is because if we make a single cut to generate so-called indel mutation to basically disrupt the reading frame, that won't work for non-coding RNA because they are insensitive to the reading frame. So for that, we offer 2 strategies to solve the problem. One is to introduce a paired sgRNA, we call pgRNA, to generate deletion in the genome in a high-throughput fashion to decode the non-coding RNA function. So we can use this kind of strategy for the first time to understand the essential lncRNA. And some of the lncRNA, if we knock it out, they will basically make the cell grow much faster, okay? But this kind of strategy is very useful in a number of other applications. For example, we can simultaneously knock out 2 genes, okay, to study gene and gene interactions, to understand synthetic lethality, those kind of problems. But technically, it's kind of challenging to scale up. So that's why we developed an alternative way to do the kind of deletion kind of screening to perturb the splicing. So that way we can either get exon skipping or intron retention. So that way we can really easy to scale because we can come back to using single guide RNA to make a gene deletion or intron retention. And this is just an alternative way to study lncRNA. So for deletion strategy, as I said, we can also apply to understand gene interaction. And sometimes we can use the same strategy to make a deletion to understand special structure on the genome, like TAD, the very special kind of structure to regulate the enhance and promote the kind of long-distance interactions. So in collaboration with one group from UCSD, we study a very special kind of a 3D structure on the genome called hub or hotspot. So this very unique structure basically is an interaction of one spot on the genome with multiple sites on the same genome, okay? So the hypothesis actually, they might play a certain role because their existence and also the location and number and specificity for this very unique kind of structure varied from cell to cell, from tissue to tissue, okay? So through this kind of collaboration, we basically figure out, actually demonstrate certain this kind of structure play such an essential role actually to regulate the essential gene or essential pathway from long distance. So basically, it's not because the very structure, they encode certain essential gene, rather they regulate essential gene or pathway from long distance through 3D structures. Regardless it's the protein coding gene or non-coding RNA, we all want to understand the detailed mechanism after we dig out or we screen out certain important gene, protein coding gene or non-coding RNA. And to understand the kind of mechanism, we need a very fine mapping or scanning methodology to understand the detail, even the residues in the proteins, how important it contributes to the protein functions. For that, we derived a new way to do the protein scanning. We call it PASTMUS, okay? So here's the example for PSMB5, which is a melanoma cancer drug target, okay? So for drug-resistant kind of scanning, we can identify a number of residues at a single amino resolution. We can basically identify and investigate and find out all those color-coded amino acids. Their alteration will confer drug resistance, okay? So it's very easy and very straightforward to understand this very small molecule. The pocket surface amino acids has been identified through this kind of scanning. But you can see clearly, after the pocket, we can still identify a number of other important amino acids. Their alteration will also contribute to drug resistance, okay? So this is protein scanning kind of strategy to offer the functional relevant residues. And of course, we can also turn this into a protein evolution way to evolve A protein to B proteins. In the pandemic, we also initiated a number of screening strategies. One of the strategy basically to identify so-called alternative strategy, alternative way for SARS-CoV-2 infection. We all know ACE2 is a major dominant receptor for SARS-CoV-2. And we have a good reason to argue and think there must be existing of alternative pathways. The reason is because the ACE2 expression in a number of tissues is very low or even no expression. And clinically, results tell us the viral can almost invade any kind of cell. So through this kind of gain function screening, we can identify 3 new receptors, and all of them interact directly with N-terminal of spike protein of SARS-CoV-2, unlike ACE2. Direct interact with the RBD domain, which located at the C-terminal of a spike. And so this becomes such a powerful way to screen for interesting, important protein coding gene and non-coding RNA. But nowadays, we switch a lot of effort in high-throughput screening directions to study directly the residues, okay? And the idea actually is many of the alteration of the residue located in the genes, actually, they will generate gain function phenotype, which is completely missing if we only use gene as a unit for the targeting, okay? So the first try, we choose lysine coding gene, codon as a first initiate test. The reason is because the lysine is a very popular site for post-translational modification. It's heavily related to the protein function, okay? Indeed, we can design almost more than 1/3 of all the K codon in the whole protein, in the whole genome. And this is over 200,000 of K sites can be designed, including this very screening effort. And then we indeed can identify a huge number of K sites, their mutation will give us a very strong phenotypic change. Okay, in this very screening, we use fitness, cell fitness as a readout. So basically, we can identify two groups of K mutation. One will lead to cell death, and another will promote cell growth. And very interestingly, many of the identified K mutations, okay, pro-cell growth or lead to cell highly proliferate kind of a consequence. They are not existing phenotype change if you knock out the gene or overexpress the gene itself. So it's typically a gain function. So because of time, I'm not going to get into any of the details. Nowadays, we also finish the whole screening on STY3 amino acids. They are phosphorylation sites. And you can see we can identify many of the new STY mutations. They will confer a number of, not confer, they will basically give rise to cell proliferation gain and clearly associated with the oncogenic kind of phenotype in a clinical database. So I will quickly switch gear to the gene editing itself. Okay, if we think about it, we have a number of choices of gene editing, and we all want to directly apply this into therapeutic kind of application regarding what kind of way we can use this very tool. So two directions. Oftentimes, we pay much more attention. One is we want to have as high efficient as possible the tool. And at the same time, we want to, the kind of therapeutics is as safe as possible. If you think about it, all the mechanism, all the technical advancement is what we learn mostly from bacteria, okay? Besides, addition to zinc finger kind of approach, the tail or CRISPR system is all the kind of trick we learn from bacteria, okay? So that's why we have a number of problems associated with very origin of the finding. One thing is we might have the immunogenicity problems because of the way and the proteins we learn from bacteria. So this is regarded as a foreign protein we have to deliver into human cells, for example. And of course, we'll suffer from a lot of other issues like off-target problems and the delivery kind of burden. So when we think about it, okay, we thought we might have an alternative way to learn from eukaryotes or even human cell itself, okay? And typically for gene editing is we have existing on-editing mechanism. One is ABAR, which can turn C to U through the deamination kind of activity. And another ADAR family proteins, whether it's ADAR1 or ADAR2, can convert A to adenosine, okay? So because adenosine basically with C is like A to G kind of editing. So we make it happen and develop a new way to do gene editing. We call it LAPR, short for leveraging endogenous ADAR for program editing RNA. So basically, we figure out how to send a long antisense RNA into cells. They can basically form base pairing with the target transcript. Of course, we engineer the AC mismatch to favor the A targeting, okay? So we can demonstrate this very technique. Besides with a pretty precise and high efficiency, it's not pretty safe, okay? First of all, we only have to send in a special design we call ARNA, which is ADAR recruiting RNA into the cell without sending any foreign protein, okay? Not to mention, we don't need the CRISPR system as a delivery tool, okay? So this very ARNA, after sending into cells, will not interfere the ADAR function for native site, okay? And secondly, after we send in the ARNA into cells, we didn't notice any of the global transcription change, okay? Not to even including the target gene, in this very case, PPIB. And thirdly, we didn't introduce any of the noticeable innate immune response, okay? So of course, it's only in the cell level. So one demonstration for application of LAPR is we can correct certain disease, which is typically G2A mutation generate premature stop codon. So this case of Hurler syndrome, basically this very enzyme is very important for the degradation of GAG in lysosome, okay? because of the G2A, actually rather G2A mutation, to have complete loss of gene expression and loss of enzymatic activity. So clinically, it's very bad disease, and the children suffer from this kind of, especially homozygous knockout, cannot survive past the age 10. So we're lucky enough to get the primary patient sample to test this idea. So you can see clearly we have different design of LIPR ARNA, and at least one of them actually can completely to recover the loss of enzymatic activity from zero and even past the heterozygous primary patient sample activity. So demonstrating a very exciting therapeutic potential. So back then, at the first time, at the very beginning, we realized we can do this LIPR RNA editing in a reverse way to use mRNA, internal mRNA, treat as aRNA to basically edit the reporter RNA. So that way we can basically monitor the RNA abundance. We do see the signal, but it's pretty noisy. The reason is because we don't want to overexpress ADAR proteins, okay? So kind of hold that kind of project. Last year's 3 papers basically make it happen. Of course, they all have to overexpress ADAR to boost the signal, okay? We are very excited to see this become a reality because of the potential huge application in this very direction. Basically can either monitor as a so-called RNA sensing or cell typing. And we have also have a very exciting idea how to turn this into therapeutic application in the future. So what we were doing actually at that time is to try to optimize LIPR technology, okay? Again, for 2 directions, okay? One is efficiency. We want to increase efficiency as much as possible. And at the same time, we want to solve a very special type of so-called safety problem, which is a bystander off target. The reason is because ARNA, the LIPR technology, we send in pretty long antisense RNA. So although we have ACE mismatch to dictate what kind of, which adenosine being added by the LIPR, but you can see we have a number of A in the target messenger RNA. Although we have AU perfect matching, which is not favored for ADAR to do the acting work, but we cannot completely eliminate this kind of unwanted off-target. So for these 2 kind of pursuit, we basically developed a new way to do LIPR. We call it 2.0, upgrade version. So I just want to use one slide to summarize what we have been doing. So to increase the efficiency, we try a number of ways. But essentially, we decide to choose a circularization of ARNA to make linear ARNA into circular. So that basically gave us a much more stable ARNA present in the cell. And consequently, we see the dramatic increase of editing efficiency. At the same time, we see, surprisingly, very happy to see the prolonged editing window, okay, because of the circularization of ARNA. So for the unwanted bystander off target, we figured actually if we delete the nucleotide in ARNA opposite to the A, which we don't want to edit, okay, we can almost completely eliminate the editing on this A. So both ways we can combine nicely together to have a 2.0 kind of version to do RNA editing. So basically, I just want to briefly mention, we already apply in a number of disease models. So in this NHP monkey model, you can see the editing of LIPR technology through AAV delivery can last at least 13 weeks. And we anticipate this should last a much longer time because we have to sacrifice the monkey to collect the data. So another effort is to try to turn this into another very important application to perturb splicing. That way, we can really tackle a number of other problems, especially in the way of DMD, muscular dystrophy problems. And a popular application is to skip certain exon which contains the mutations. That way, we can recover the full function of dystrophy. Okay. So basically, to I will quickly summarize because of time, and I'll try in a disease model in monkey. And you can see the editing can last close to 1 year. And phenotypically, you can see we have wild type monkey. You can see clearly the dystrophin expression and the localization on the cell surface, okay? And together with another very important proteins, team up with dystrophin labeled in red color. In the DMD model, disease model, you can see almost dystrophin proteins. The green flash is completely gone, okay? And the syntrophin can no longer locate to the cell surface, okay? By the treatment using the LIPR, AAV delivered LIPR, you can see the recovery of both dystrophin and the syntrophin can last for 12 months. Okay, so that's a pretty exciting result. So because we play with the circularizing of ARNA, so during the pandemic, we turned this into a protein expression platform, and we demonstrate using circular RNA as expression platform, we can really generate high efficient protein production to make the very effective vaccine. And we can demonstrate in both mouse and NHP model can really provide protection against the real viral infections. And I'm happy to say we already entered the clinical stage, and so far we have a pretty decent safety result, and also even after first dose, we already see the very significant antibody boost from the patients. So lastly, I'll spend 1 minute to talk about our recent development new tool, which is base editing in mitochondria. As we all know, we have a number of mitochondrial-associated specific disorders, and most of them is because of a single point mutation. So that's why it's hugely demanded actually to have a base editing technique targeting mitochondria. Okay, but unfortunately, the only way to do base editing is based on CRISPR system, okay? But CRISPR system so far cannot work in mitochondria because of difficulty to get the RNA into the mitochondria. So if we come back to the protein targeting of genome editing kind of a version, which is a tail or zinc finger, unfortunately, they cannot do both editing, base editing. The reason is because they don't have the capability to unwind double-strand DNA to allow single-strand DNA deaminase to work, okay? So until 2020, okay, David Liu and his collaborators to figure out how to utilize a very special type of deaminase, which is a so-called double-strand deaminase, DDDA, okay? They make the work in mitochondria because they can combine with the tail system, okay? But the problem actually for this kind of strategy is because its DDDA can directly interact with CTCF, cause global off-target in the nucleus genome, okay? So we think differently. We try to think about this very problem is we want to utilize the commonly present single-strand DNA deaminase to work in and to work with tail to make a base editing work in mitochondria. To do that, the only thing we think is if we can generate single strand, we might have chance to make it work, okay? So in this very case, if we do A to G kind of base editing in mitochondria, you can see if we use a pair of tail to introduce TADA, which is the A to G kind of deaminase, you barely can see the base editing work. If we combine with mut-H with the nickase, can really make a nick on double-strand DNA. That way, we figured we will have at least transient single strand present, okay? And you can see clearly we can generate base editing when they team up with TADA. And we can demonstrate that nickase activity is essential because if we create a mutation to disrupt the nickase activity, the editing is completely gone. So that way, we can easily to do G to A kind of version, A to G rather, and also C to T because we can switch the TADA with alpha-back system. You can see we can have another version of a base editing. And we can demonstrate this very version is much safer if you compare with the DDDA kind of version. And we have very clear background in both mitochondria and the nuclear, and the efficiency remain pretty high. And using this very new technology, we can easily create disease model, okay? And again, I want to emphasize, to do that, we use circular RNA to really significantly boost efficiency to generate the mutation, to have this disorder of mitochondria, because we can generate a specific mutation in certain important gene. We can also correct certain point mutation, and this is a very famous LHON problem. We know exactly what kind of mutation, single point mutation, occurring in what kind of gene, and we can really make a conversion to largely recover the lost and defective function for mitochondria, demonstrates the therapeutic potential of this very disease. With that, I want to close my talk by acknowledging my group, my many collaborators, and many of them sit in the audience, and my funding support. All right, thank you very much.